Then we have psychological recording. No, we have first histological methods, then we have psychological recording. So with the advent of technology in the present time, there are many techniques and methods to study anatomy and functioning of human brain. For studying minute details of tissue like microscopic investigation, histological methods are used for this purpose. In this method, the sample of the brain that is under investigation needs to be prepared so that it can be mounted on a slide and observed under a light microscope. The neural tissue is very fragile and under normal condition it may just disintegrate. Hence, the tissue needs to be fixed and stained in order to be viewed on the slide. When it is taken out, the bacteria or molds tend to decompose the neural tissue. So, in histological methods, we will study the anatomy and functioning of human brain. Okay, and for studying the minute details of tissue, we do microscopic investigation through histological methods. Okay, in this method we just take a sample of the brain which is under investigation, and we will stain it, then we will mount on a slide and observe it under a light microscope. Okay, so let us now look at the procedures in the histological methods. Like fixation, in this method a fixative is used to fix the tissue. The tissue is cleaned and dipped in fixative solution. The most commonly used fixative solution is formalin. The fixative prevents autolysis and makes the tissue hard, but still the tissue is not so hard to be cutting using a microtome. Microtome is automatic slicing machine used in microscopy to produce thin sections of the fixed and embedded tissue for microscopic observations. Hence, the brain tissue is freeze so that tissue becomes more hard. It is also used to stabilize neural tissue. Autolysis. What is autolysis? Auto means self. The autolysis is the self-digestion or the breakdown of the cells in animal, plant, or microorganism tissue by their own enzymes. Then the next procedure we have freezing. First we did fixation. Now we have freezing. In freezing, the tissue needs to be frozen in order to be sliced. To freeze the tissue, it is dipped in a sugar solution and then put in a freezer. Okay. The temperature is regulated in a way so that the tissue is maintained at an appropriate temperature. Once the tissue is hard enough, it is to be placed on the microtome to be sliced. So this was the second. What was the first? First was fixation, freezing. Now we have embedding. Another way to make the brain tissue hard is by embedding and covering it by paraffin or nitrocellulose. Once the tissue is hard, then it is cut using a microtome. These slices are then placed on a slide and mounted on it using albumin, which is taken from the egg. Embedding is the covering it by paraffin or nitrocellulose. The slide is dried. It is then dyed using different chemical solution. Embedding is the other way to make the tissue hard by using paraffin or nitrocellulose. These slices are then placed on a slide and mounted on it using albumin. Albumin is taken from the egg. Then the next procedure we have staining. It is done so that the structural details of the tissue become apparent under the microscope. This is done by darkening or coloring particular features of the tissue. Nasal stains or cell body stains are most frequently used to dye the cell bodies. Myelin stain colors are the myelin sheet to study the nerve pathways. Membrane stains like Golgi Cox stain helps to color the cell body, dendrites, and the axon. So here we have membrane stain like Golgi Cox stain, which helps to color the cell body, dendrites, and axon. And myelin stains color the myelin sheet, which is the part of nerve cells. Nasal stains or cell body stains are most frequently used to dye the cell bodies. And we have psychophysiological recording methods. In the psychophysiological recording methods, we have EEG. and magnetoencephalography, electromyography, electrooculography, and the electrodermal activity. Okay, so the first is electroencephalography. The electrical activity of the brain is recorded using electroencephalograph machine, and the technique is known as electroencephalography. Electrodes are placed on the scalp of the patient and recording is done on the computer. Earlier, a rolling paper was used where the readings were displayed. Now, a computer printout is taken. The EEG signals helps in the diagnosis of various disorders of the brain, mild head injury, cerebral pathology, epilepsy, and speed of processing information on how the memory functions with an increase in age. The EEG signals help in the diagnosis of various disorders of the brain. So, in this, the lectures are placed on the scalp of the patient basically, and the recording is done on the computer. Earlier, a rolling paper was used where the readings were displayed. The computer printout is taken. And next we have magnetoencephalography, which is also referred as MEG, which measures the changes in the magnetic fields on the surface of the scalp produced by changes in the basic patterns of the neural activity. In MEG, we we notice the change in the magnetic fields on the surface of the scalp, which is produced by the changing in basic patterns of neural activity. And then we have electromyography. The muscle tension is recorded with the help of electromyogram. The electrodes are attached to the skin over the muscle that is to be investigated. When the muscle contracts, producing muscle tension, it is recorded. This information is the basic which help us to know about the muscle tone. So in the electromyography, we record the muscle tension, and the electrodes are attached to the skin over the muscle that is to be investigated. Okay, and the next we have electrooculography. It is the eye movements are recorded by using an electrooculogram EOG. There is a slight difference in the electrical potential in the back and front portion of the eyeball. This difference is potential is recorded with the help of electrodes that are placed near the eye. Okay, like in this, the eye movements are recorded. That's why its name is oculography. And the difference in potential is recorded basically with the help of electrodes that are placed near the eye. Next, we have electrodermal activity. Here, two main indices of the electrodermal activity are skin conductance level SCL and skin conductance response SCR. Electrodermal activity of the skin is recorded by attaching the electrodes on the fingers and recording the fluctuations in the skin conduction. Electrodermal activity of the skin is recorded by attaching the electrodes on the fingers. Okay, we measure the skin conductance by attaching electrodes on the fingers and recording the fluctuations in the conductance level. So SCL measures the background level of skin conduction associated with the particular situation, and SCR is a transient change in skin conduction associated with the distinct experiences. SCL is the particular situation conductions and the SCR is the distinct experiences. When there are different kinds of emotional stimuli, variations are produced in the skin conductance level, which is SCL, due to changes in the sweat gland activity.